Okay, hi everyone. Um, let's continue talking today about the role of RB in controlling this family of transcription factors, the E2Fs. We had previously introduced RB in our discussion of retinoblastoma, right? So that's the tumor or the cancer disease from which this RB gene was first identified. And I've already introduced that the way RB works is somehow by controlling the activity of transcription factors that, if these transcription factors are allowed to do their job, would actually encourage the cell to move forward in its cell cycle. So that's why RB acts as a tumor suppressor protein, right? Because it represses or suppresses the cell's desire to leave G1. When we talk about intracellular signaling pathways, I think one thing that often happens is that students get very confused. You know, you have these pathways and arrows are pointing at different things, and proteins are communicating with other proteins, and it can get kind of overwhelming, right? Because you lose track of the big picture of what we're talking about. Right? So one way I like to break this down is to emphasize that whenever you're talking about a chain of different proteins taking turns interacting with each other, the reality is each individual interaction between one protein, right, and another protein itself is not some crazy, hard to understand process. Right? Each individual interaction in a chain of proteins that make up a pathway in the cell is in fact a very simple event. So this type of a cartoon here, right? I used to love these as a kid. You know, you've probably seen something like this before, right? This is a Rube Goldberg drawing, and so he was very famous for making these fanciful kind of imaginations of how you could accomplish very simple things through what seemed like an overly, overboard kind of method, right? So here, what he's showing is that you've strung together a whole bunch of contraptions here, right? And this actually represents his sort of fanciful orange juicer, okay? So what you see is that it starts with step A, which is an empty glass milk bottle left on the windowsill. You know, this is pretty old, right? This is back in the days when people didn't actually buy milk from the store and put it in their fridge, right? You'd actually have fresh milk delivered every day, and you'd put the empty milk bottles out so that the milkman can take the empty bottles back. So when the milkman comes and pulls the bottle, right, it pulls this pulley here, that causes this sword to swing, and it cuts the rope that allows this guillotine blade to fall, that then releases this log that's been sort of chained to the ceiling. And the log then swings over and knocks this door closed. Right? And so when the door gets knocked closed, there's a sickle, right, a sickle on here that will then slice the bottom of this orange that's hanging there. At the same time, the door up here, right, has a little, I don't know what this is, a little spike, right, that sort of jams into this poor parrot up here, causing the parrot to freak out, and the parrot squeezes or pulls down on this lever, and that causes this boot to fall down and squash this octopus. The octopus gets really mad, right, and wants to then go ahead and grab and squeeze something, so it grabs the orange, and it just squeezes your nice glass of fresh orange juice from this. Right? Now you might think like, okay, yeah, that's kind of fun and kind of crazy, but what's the point? The point is you can think about this as an analogy for the series of events that happen when some sort of extracellular mitogenic signal arrives on the outside of the cell membrane of a cell, right, such as an EGF that gets received by a receptor tyrosine kinase like EGF receptor. Ultimately, a series of events are going to happen that represent that signal transduced from the cell membrane into the cytoplasm. And you'll have multiple proteins that then sequentially talk to each other, right, resulting in those signals arriving in the nucleus. And ultimately, the squeezing of the orange to get your orange juice is the gene expression that results. But the critical thing I want you to think about here is that each one of these little events that happens, labeled by a letter A through O in this Rube Goldberg fanciful cartoon, is itself a very simple action, right? Like the log hitting the door, there's nothing fancy about that at all. Right? So I want you to start thinking about this as how proteins interact with each other, because the interaction with one protein with another protein is itself can be a very simple event. Right? It's just when you chain together the whole series of simple events, amazing, complicated things seem to happen. So let's look as an example of mitogenic pathways signaling through, for example, like RAS. Right? So we said that as part of the cell cycle regulation, the cell cycle is controlled by the rise and fall of cyclin protein levels. So for example, here right in the middle is cyclin D1. Right? So let's say this is the cyclin D1 gene that codes for cyclin D1. So stimulatory activity that kind of arrives at this gene, in this illustration, we can think of as actions that result in that gene then being expressed and cyclin D protein levels going up. Right? So during the start of G1, you'd have very little cyclin D1 protein in the cytoplasm, but right, the cell starts listening to signals coming in from the outside. Remember, it's only during G1 that the cell is responsive and listening to external signals, controlling things like cell proliferation. So let's say growth factor gets received by EGFR, and it ends up activating RAS, right? And so here we have then the message that's arriving on the surface of the cell being interpreted by tyrosine kinases, right? And that's what gets the signal into the cell. Right? Then RAS, right? Because RAS is a very important supervisory type of proto-oncogene, right? It's in charge of now potentially talking to multiple pathways of different proteins, right? That all sort of result from RAS being in its GTP bound state and being able to talk to these. So RAS talks to three main pathways that we looked at: RAF pathway, the MEK pathway, and the GSK 3K pathway that controls things like beta-catenin. So you can think about that as like this log hitting the door, because remember that log that hits the door represents a divergence in the pathway. So not only does the door have the sickle that cuts the orange to get the orange ready, but a separate pathway also emanates from that same action of the log hitting the door, which is the parrot getting upset and then causing the boot to fall on the octopus, right? So here in the cell, you would have a divergence of different pathways. These pathways then, right, with one protein like a kinase phosphorylating another kinase, which then targets another kinase, each one of these little interactions is a very simple event, right? Two proteins come together and they're the right shape so that one passes a phosphate group to the next, right? Just like the parrot or the boot or the octopus by itself doesn't sound necessarily remarkable or complicated, right? But ultimately, these signals converge on the nucleus and cause an increase in the expression level of target proteins like cyclin D1. So if you think about this in the context of the cell cycle then, right, all of these signals that are arriving at the surface of the cell during G1 are doing things like controlling whether cyclin D1 levels are rising, right, so that cyclin D1 can then pair up with the cyclin-dependent kinase proteins like CDK4 and 6, that then together, the cyclin D1 helps change the shape of that CDK. So now that CDK can talk to different other proteins and kick off other pathways, right? If that mitogenic signal no longer is present coming in, right, then maybe cyclin D levels will be sort of temporarily halted or might actually go down. So that's why during G1, there can be sort of back and forth, right, about what the cell is doing, because depending upon the dynamic nature of signals coming in, right, that can keep the cell sort of in G1 going a little back and forth, just doing its job. But If you keep getting strong mitogenic signals, right, then instead of the G1 kind of going back and forth, what happens is you're always going forward through the G1, and then eventually you get to that restriction point, right, where the cell suddenly then plunges forward into the cell cycle and then goes into autopilot, right? Because as we learned last time, once the cell gets into S phase, it's committed to go all the way through to mitosis, right? It cannot back up again into G1, right? Even if you took away all those mitogenic signals, right? Because at that point, it's no longer listening to what's going on, what it's being told from outside. It is on this autonomous program of just the different cyclin levels with CDKs driving waves of protein expression that ultimately drive things like DNA replication, stuff like that, but also the production of the next wave of cyclin and CDK proteins that will be needed to push into the next cycle. So we looked at this last time, right? And we said importantly, right, that the expression in waves of these different combinations of cyclins and CDKs is what moves the cell through the cell cycle, because each combination of these, right, controls a different set of proteins to activate, ultimately targeting a different set of genes to be expressed. And because of that sort of ratcheting shape of the expression levels of cyclins, that's why the cell cannot back up. I think last time I mentioned to you, it's kind of funny to think that the cell can't go backwards in the cycle, right? I mean, obviously, it's good that it cannot, because that would really mess things up. Can you imagine if a cell got halfway through the S phase and it made, let's say, 3/4 of a duplicate of the genome, and then for some crazy reason, it just decided, you know what, I'm not into this today. I don't want to do this anymore. And it decides to go back into G1. It goes back to G1 with like a totally weird, messed up ploidy, right? That would be really bad. But yet, if you think about it, right, the cell doesn't actually know what it's doing or whatever. It's all driven by just proteins interacting with each other. So in a scary kind of way, what is to prevent a cell from backing up if it's just proteins interacting with other proteins? It's because we evolved this rigorous system of depending upon these cyclin waves to be the things that control moving forward and not being able to back up, right? That's kind of cool to think about. I think that's really cool. Right. So we did mention that in late G1, right, if the cell keeps getting mitogenic signals, at a certain point, the buildup of proteins like cyclin D and CDK4, right, is starting the production of other proteins that might be more relevant for S phase, right? So those are building up. And importantly, throughout G1, as you were getting more cyclin D, CDK4/6 activity, one of the other target sets of genes based upon that are the cyclin E and CDK2s, right? So those levels of these purple cyclin CDKs are building up as well, right? And so once you cross that R point, then right, these cyclin E CDK2s take over and start driving the cell to really cross over into S phase. So this R point then is a very important checkpoint because once the R point is crossed, the cell is committed to finishing this cycle, unless problems get in the way, at which point maybe the cell needs to stop. And if it cannot fix those problems, the cell just has to give up, right, rather than continuing. So how is this critical R point guarded, right? Because obviously this R point is very important. So that's the job of RB. Right? So now let's talk a little bit more in depth about RB. We looked at this slide before when I first introduced to you how RB works. So RB's job, so here's the RB protein, which obviously is encoded for by the RB gene. It sequesters or prevents a family of transcription factors known as the E2Fs from being able to carry out their transcription factor function. It does so because RB physically binds to, and in some ways you can think of it as covers, the E2F shape structure so that the E2F can't function. Right? If RB is phosphorylated, right, then the phosphorylation of RB can result in the shape of RB changing in a way that it then lets go of the E2Fs. And then the E2Fs are allowed then to bind to their DNA binding motifs in promoters and enhancers of different genes, right? And then those genes start being expressed. And of course, those genes are the ones that represent the next wave of proteins that need to be made in order to get the cell ready for something like S phase, right? Including things like the cyclin E, right? So what phosphorylates RB? RB is phosphorylated by the CDK4 cyclin D complex, right? So as you get more and more cyclin D building up in the cell as the result of things like growth factors continuously stimulating receptor tyrosine kinases to turn on RAS or whatever, right? So on that previous slide, we saw how many pathways converge on cyclin D1 expression. Right? So the more of this CDK4 cyclin D1 you have, the more of the RB molecules in your cell are getting phosphorylated. So the more they are letting go of the E2F transcription factors. On the RB protein, there are 16 potential phosphorylation sites, right? 16 spots on the RB protein that we now know have the potential to have phosphate groups added to them. As a result, that gives you a lot of sort of variability to play with, like 16 levels of phosphorylation, right? So that means a given RB protein molecule might be completely unphosphorylated, or it might have a low number, right? A few of these sites phosphorylated, in which case we might call it hypophosphorylated. Right? Or it might have many or all 16 sites phosphorylated, in which case we would say that this RB is hyperphosphorylated. Okay? So that's very important, because if you look at the status of RB protein phosphorylation as you move through the cell cycle, you see something really remarkable. Right at the start of G1, let's say that this is a daughter cell that has just finished separating, right, from the parent cell that successfully went through the cell cycle, went through mitosis, and now you have these two daughter cells, and each one of these daughter cells now is starting from square one, right? It's now ready to get away from thinking about mitosis, right? It now wants to resume its normal cell functions. So at the beginning of G1, you would have very low levels of RB phosphorylation. In fact, if the cell were to take a break and go off onto the little pit stop, the little rest stop, and say that it went into G1 arrest, during G1, nearly all of the RB protein is unphosphorylated. Right? But let's say the cell is in G1 and it's moving forward. Right? As the cell G1 moves forward, right, and maybe it's kind of backing up, going forward, backing up within G1, based upon what signals are coming in, you would imagine more and more RB gets phosphorylated, right? So as you're moving through G1, the RB is in a hypophosphorylated state, right? Out of its 16 possible phosphorylation sites, you'd find sort of a smattering, right, of some RB proteins having a few sites phosphorylated. This hypophosphorylation is actually very important, right? Because the proper shape of RB, in order to do its job, actually does depend upon a couple of its potential phosphate sites having phosphate groups on them, right? So throughout G1, RB is hypophosphorylated. But what you see is right at the point where the cell is approaching the restriction point, right? You'd find more and more of the RB protein molecules are now hyperphosphorylated, right? And so when they're hyperphosphorylated, that's the shape when they no longer can hang on to the E2Fs, right? And the E2Fs transcription factors are released, and those transcription factors then go to work. So that's why during the first part of G1, the little red line that shows the amount of RB phosphorylation is very minuscule, but then very quickly in G1, right, there's sort of this low level of RB phosphorylation. RB is hypophosphorylated, but then once you get past the R point, for the whole rest of the cell cycle, look at this big thick red line, right? For the whole rest of the cell cycle, RB is hyperphosphorylated, right? So S phase through mitosis. Because, right, for the whole rest of the cell cycle, you do not want RB protein, right, messing things up by interacting with transcription factors, right? Because if the cell at this point is on autopilot and it's depending upon these different waves of cyclins to carry out these pre-programmed series of events through S phase and G2, you don't want RB in there kind of like, hey, let me hang on to some of these transcription factors and slow things down, right? So that's why RB remains hyperphosphorylated all the way to the end, right? And then right at the completion of mitosis, remarkably, in a very quick wave of activity, all the RB gets dephosphorylated, right? All of that phosphorylation gets erased. And so the daughter cell that emerges starts again from unphosphorylated RB. So you might say, wait, why go to the trouble of just keeping RB around, but in a hyperphosphorylated state? Why doesn't our cell just go ahead and get rid of the RB protein, right? It's because you need to have a lot of the RB protein ready to go right at the start of G1, right? And remember, through G2 and mitosis, you're not doing normal gene transcription. So it's not like during mitosis, you can be busy trying to make more RB because you got rid of it all earlier, right? No, right? You cannot, because that can only happen really during G1, but during G1, you need to have the RB there. So the way we evolved is we keep the RB protein around through the rest of the cell cycle, but just make sure that it's turned off through hyperphosphorylation. And that's why it's neat that the moment mitosis is done and you need RB to be freed up and ready to start going again, right? You just dephosphorylate all the RB. So let's look at it this way. Here during early and mid G1, right? Early G1, you'd have unphosphorylated RB. So this green thing is the RB protein, right? But then as you start getting activity of cyclin D and CDK4 pairing up together because mitogenic signals are arriving at the cell, right? Those start to hypophosphorylate RB. And so it's this hypophosphorylated RB that does a good job then of sequestering or hanging onto the E2F transcription factors. But as you move through G1, again, just as long as the cell keeps receiving more mitogenic signals, right, compared to inhibitory signals, then over time, you'd get more and more phosphorylated RB. And ultimately, right, when you cross this R point, what you find, or what I should say, is that once you have enough hyperphosphorylated RB, you then cross this R point, right? And then you have the also added activity of the cyclin E and CDK2s that are building up, really contributing now to the hyperphosphorylation of RB, right? Oops, sorry. So let's look at some ways that RB function can be impaired. We already looked at, some obvious examples of how proteins can lose their function through mutations, right? So obviously, if you mess up the amino acid sequence of the RB protein, you might have an RB protein that doesn't function right, or maybe isn't even made anymore, and that could have really disastrous effects on the cell's control over cell cycle. That's why when we looked at the retinoblastoma disease, right, we said that inherited defects in the alleles of RB can result in inactive RB in one copy of the RB in the genome, like the one that a child inherited from mom, right? But just as long as a cell still has a good copy of the RB gene, like the one that came from dad, right, then that cell is still making usable RB protein, and that can still keep the cell cycle from moving ahead out of control, right? But once you lose that other good copy in that cell through somatic mutation or some sort of translocation event, then that cell has lost RB and lost the control that RB has over the cell cycle, right? But there are other ways that RB function can be lost as well. And this brings us back to our discussion about tumor-causing viral genes. Right? Remember, we said that many of the genes from viral genomes that stimulate transformation of cells have nothing to do with the actual production of viral proteins, right? They have to do with trying to encourage the host cell to shift out of G1 to be in that state that's ready to do things like DNA replication, right? so that it can then start making viral gene products from those other viral genes that actually encode for components of the virus. So we had talked about early genes that are transforming versus late viral genes that are more replication genes, right? And we said that many of these early genes are the fragments that you find when you actually take biopsies of aggressive tumors like cervical cancer, right? And then you can find evidence of these early viral genes in them that transform things. So we talked about that a lot. Let's take a look at how the products of these early genes actually do this, right, and transform the cells. So some examples of these genes are things like viral gene EA1, large T antigen, and E7, right? So these are all oncogenes. And the interesting thing about this is these are genes that do not have a normal proto-oncogene corresponding gene in our genome, right? So these are examples of genes that were cooked up just through mutations in viruses over time, right? So our human genome doesn't have large T antigen or E1A or E7. So what do these proteins do? Here was an early experiment where they looked at trying to identify what types of factors are coming from these tumor-causing viruses that are responsible for transforming cells. So these are two different cell lines that they're comparing. So one set of cell lines is a cancer cell line. These are HeLa cells that were originally turned into cell lines starting from HPV transformed cells, back in the 1950s, right? And to this day, the HeLa cells that we use, right, no longer actually express those original transformative genes that were introduced by the virus into them. They've had so many mutations that they just continue to be aggressive tumor cells. This other cell line here, this 293, so 293 cells are a epithelial cell line or fibroblast cell line, I think, where researchers can transform them by putting viral genes in them. Okay, so these are adenovirus transformed cells. So what they've done is they've taken these two cells, they've taken the cell lysate, all the proteins out of them, and they want to examine using Western blot. Are there any differences in the types of proteins that are being expressed in abundance in these two types of cells? Right? Both of these are transformed cells, but one of them was a cell that was transformed a long time ago, the HeLa cells, and now they don't really show any evidence of any viral proteins. This other one, the 293, was one that the researchers just kind of transformed themselves right now with virus. Right? So this is what they see. The purple column, the C36, right, is probing the Western blot using an anti-RB protein antibody. Okay. This other column, the M73, is probing the blot using a antibody against early viral oncogenes like the E1A. Right? So you can see in both of these cases here, right, the HeLa cells, right, and the adenotransformed cells, when they looked for the presence of the E1A viral gene, as I mentioned before, in the HeLa cells, you no longer detect it, right? So the right column, you don't see any evidence that there are those transformative E1A viral proteins in it anymore, right? Whereas if you look in the 293 cells that the researchers just transformed with these viruses, not surprisingly, you see the E1A protein here. Okay. So what they noticed was that in these cells, both of these still make the RB gene, right? So that's what this is here. This P105 is the RB protein, right? Or I shouldn't say they make the gene. They obviously still have the RB gene. They make the RB protein. So that's what this band is here, right? So even in the adenovirus transform cells, they make it as well. But what they noticed is with the abundance of the E1A protein, right, you get less of the normal RB right, and much heavier levels of these other larger forms of essentially RB. So like the 107, 130, these are RB that essentially now, right, migrating slower on this Western blot. And why do you think they'd migrate slower? They migrate slower because they're heavier, because they are bound up by things like some other protein, right? Okay. So for example, what they're looking at here is if you have the antibody against the E1A, so that's what this green thing here is, right? So if there's the E1A protein present in the cell, this brown thing, it will bind to it, right? But it will also bind, right, to anything else that it happened to be attached to inside the cell. And what they realized from the data is that it looked like the E1A protein is binding to the RB, and that's why the RB looks heavier. It looks heavier in the viral transformed cells. In addition, you can see here, right, you can detect that it's also bound to this heavier form. P300 is a histone acetyltransferase. So histone acetyltransferases, modify chromatin. And so you can tell that E1A is also apparently bound up to the P300. So for our lesson today, I want to focus on this observation. The E1A is actually bound to the retinoblastoma. So what they determined is that the E1A and E7 proteins that are coded for by the early transforming genes of the tumor-causing virus physically interfere with the ability of RB to be able to do its job of hanging on to those E2F transcription factors. If you look at the functional sort of domains of these proteins here, so these are the RB protein in the P107 version, it's sort of like a RB-like protein, and the P130, right? So if you sort of unraveled the protein and just looked at the functional domains, what you see is that part of the region in the middle here between like the A and the B region is the pocket that binds to the E2F transcription factors and is able to hang on to it, right? What they found is that the viral oncoprotein, like the E1A and the E7, bind to this very close to this region as well. So here you see a three-dimensional molecular map of this, right? So they've actually taken the green blob is sort of a superimposed structure of RB with P07 and P130, showing how similar these related tumor suppressor molecules are. And so the lighter shading means that there's more variation there, okay, between the three proteins. The darker the green means that this is a region of the amino acid sequence that's highly conserved, right? So this highly conserved pocket that you see here, right, is what actually binds to, or the interaction surface with, the E2F transcription factors, right? And this yellow squiggle is the HPV E7 early transforming gene product. So you can see, right, that protein product lays right across the surface needed for the RB proteins to be able to properly sequester the E2Fs. Therefore, if you have this early transforming protein, this peptide from the virus covering this up, right, then RB no longer works. Okay. So what about the E2Fs themselves, right? I said that, okay, the RB family of tumor suppressor proteins, they work by binding up the E2Fs, preventing the E2Fs from normally driving the cell cycle forward. So let's talk about the E2Fs. Right? So E2F is a family of 8 related transcription factor proteins. They all follow the similar types of structural domains. They all have a DNA binding domain, right? That's not surprising, right? Just like most transcription factors bind to DNA targets in order to then do other things like bring chromatin modifying transcription factors, ultimately basal transcription factors like TF2 that then recruit what RNA polymerase to the promoter. So all of them have DNA binding domains. Some of them have 2 DNA binding domains. Then you have a signal over here, the nuclear localization signal, which means this is a signal that helps these members of the E2F family actually go to the nucleus. Then over here, you see in these members over here, you have the RB binding domain, right? So this is where that dark green cleft in that RB three-dimensional model on the previous slide, right? This is where it'd be interacting with the E2Fs to keep the E2Fs from doing their job. Right? And then these E2Fs usually don't function as a monomer. They usually function as a partner with another protein. And depending on what protein that is, it might have different sorts of functions. So this is this gray heterodimerization domain that mediates that. Okay. These typically partner up with a different group of proteins known as the DP proteins. So let's see what's going on here. Normally, the E2F, right, these activated E2Fs that are allowed to kind of do their thing, right, they will partner up with a DP protein to form this functional dimer. So now this thing together is the right shape to sit on certain DNA sequences via the DNA binding domain of the E2F, recognizing certain sequences. So this might be like in a promoter of a gene or something like that. However, if you have a lot of hypophosphorylated RB around, right? Remember, hypophosphorylated RB, like you would find during the early parts of G1, that's the right shape to bind to the E2F and the DP, right? And keep the E2F and DP from working. Because the RB, when it binds, it's the right shape to recruit things like histone deacetylases, or HDACs. So not only would that prevent the E2F transcription factors from successfully signaling other partner proteins to come, but it also recruits the deacetylation of histones, right? So as the histones are being deacetylated, right, this region of the chromatin is getting more and more tightly packed up, keeping the gene expression suppressed. Right? So that's a similar story for those other RB family proteins, like the slightly heavier P107, P130. Right? They also do the same thing. However, if you go ahead and remove the influence of RB, right, so that's what this blue arrow is up here, then nothing is covering the E2F and the TP anymore. And the E2F and the DP can do their job. And their job is they recruit histone acetylases. So this red thing here. Right? And so the histone acetylase will come into this region of the chromatin, will begin to add acetyl groups to the histone proteins, therefore beginning to unravel the chromatin and making it even more and more likely that additional DNA binding domain sequences can be seen by other transcription factors and things start piling up. So normally, what causes the release of this? Well, that's what we've been talking about, right? The release of the RB from the E2F is because hyperphosphorylation of RB by building levels of cyclin E and CDK2, talking to other protein pathways that phosphorylate RB. So this would be happening as you're moving forward through G1. As you start getting kind of late G1, you have more and more of this purple cyclin E and CDK2 building up as a result of what? As a result of the cyclin D and CDK4s, right, turning on the cyclin E and CDK2 genes, right? And if you have more of this purple E and CDK2 working together, the more hyperphosphorylation of RB you get. Right? So in a normal cell, this would be due to the normal function of cyclin E and CDK2, but in a virally transformed cell, this is not happening through hyperphosphorylation. It's happening because those early transforming proteins from the virus, right, are actually blocking the interaction between the RB and the E2F in the first place. Right? All right. So I like these cartoons a lot, and I hope you guys like them too. You know, I tend to like them because I like to visually kind of see things, things that are not black and white. I mean, not like on and off, but sort of subtle, right? And that's really what I want you to appreciate. The movement of the cell cycle is determined by waves of things happening, right? So it's very subtle, very nuanced, as I like to say it. It's not really like switches are being turned on and turned off, right? So these types of cartoons, I think, make it easier for us to appreciate that. So again, in early mid G1, you have the activity of cyclin D and CDK4 and 6 together beginning to go up, right? And that results in things like the RB being hypophosphorylated, right? So just a few of its potential 16 different phosphorylation sites get phosphorylated, but that's good because that's the right shape for the RB to work, right? So it sequesters and covers up the E2F transcription factor family members like E2F1, 2, 3, whatever, right? As things begin to build up, you then get closer to the R point, right? And that means in the background, you start seeing the buildup of cyclin E's and CDK2s. Right? If I were to draw this, I would have actually put the E's and CDK2s kind of starting over here already, right? Because in the background, they are building up. And the point is the crossing over of the R point is a result of enough cyclin E and CDK2 activity to really start resulting in most of the RB proteins being hyperphosphorylated. And as a result, they let go of those E2F transcription factors, right? And now you are committed, because at this point, you cannot back up, right? And just say like, okay, forget it. You know, I'm getting cold feet here. I'm not ready for this. I just want to hang out in G1. No, right? Once you're at this point and past the R point, the cell has to go forward, because now it's going to be driven by just the successive waves of different cyclin CDK pairs. So as you move into S phase, right, now it's all about cyclin E and CDK2 signaling whatever proteins they want to signal. Cyclin D and CDK4/6 is now gone, right? And so part of that is now the E2Fs, right, themselves get shut down because you no longer need the E2Fs to push the cell into S phase. Their job is done, right? They disappear. Everything else now is just being driven by whatever's going on, by the next waves of the cyclins. And this is a reminder that from S phase through to the end of the cell cycle, you don't lose the RB proteins, right? All those RB protein molecules are still there, but they're just completely turned off because they stay hyperphosphorylated until the end of mitosis, when then in one big swoop, right, they all become unphosphorylated again. And you start from the beginning of the slide. Okay. So one thing that I want to encourage you to think about is the importance of feedback loops in driving the cell cycle forward and preventing it from backing up. When I say feedback loops, you know, what we're talking about is, if we're specifically talking about the transition from G1 to S phase, is that as you start building up levels of cyclin E and CDK2 as a result of in G1 having cyclin D and CDK4 levels up, right? Eventually, when this drives the hyperphosphorylation, right, of RB, and RB no longer hangs on to the E2Fs, the E2Fs are allowed to signal. What happens is the E2Fs will, of course, start sitting on different promoters and enhancers of a wide range of genes that are related to getting the cell into S phase, right? Like DNA replication, machinery proteins, and things like that. But E2F also stimulates more production of E2F protein. So in other words, the E2F gene itself has E2F response elements on it, right? So this is what I mean by feedback. So the more E2F protein you have, the more the E2F gene is stimulated to make more E2F protein. And not surprisingly, the genes that code for cyclin E and CDK2 have in their regulatory regions, right, E2F response elements as well. So as you get more E2F activity, you also get more cyclin E CDK2 production. So that's why all of a sudden, right, the production of these purple, if you just go by the color, cyclin E and CDK2s suddenly starts blowing up. And what does that do, right? That in a way feeds back, right? Because the more of these purple cyclin E and CDK2s you have, the more you're really making sure that the all the RB is hyperphosphorylated, right? So that's what I mean by feedback. And once things get out of control like this, right, you cannot squash it and go backwards, right? So this is like, yeah, feedback. You know, when I was a teenager, you know, I had this electric guitar. It was a Fender Strat. I loved that thing. And, you know, me and my buddies, we all wanted to be rock stars and play guitar. So I used to practice my guitar in my garage, right? Because that's the only place that my parents would let me practice it. Because I had this little amp, right? I plug it into the amp and I'd be like doing Eddie Van Halen stuff in the garage. And on this amp, it was kind of funny because this amp had different little jacks on it that you could plug things in. And one of them was the instrument input, right? So obviously that's where I plugged my guitar in with the patch cord. But there was another one that was also labeled in, like input 2 or something like that, right? And the amp had an output jack. So if you wanted to hook the amp up to a bigger set of speakers, you could also run it to those bigger set of speakers. So I always wondered what would happen if I took the output from the amp and plugged it back into its own input, right? Now, those are the type of things you think about when you're, you know, 15 and, you know, stuff like this really matters. So one day I decided to do it, right? I took my guitar cord and plugged it in the output of the amp and then plugged it into the input of the same amp. The moment I plugged it in, right, I got the loudest, craziest, most obnoxious sound you could ever imagine, right? Because basically my amp like blew up, right? Because its signal that was amplifying was fed back into itself to be amplified again and again and again. And it basically sounds like, now I know what it sounds like, what the, you know, at the beginning of each month when we have our tsunami warning tests around the island, right? This thing was like that. It was super loud. And of course I freaked out and I unplugged it. And, you know, my mom comes running into the garage, right? And she's like, what was that? What was that? And I couldn't hear her because my ears are ringing. And I'm like, what? What? So anyhow, that is the concept of feedback, right? Because once you start feedback going, right, it's very hard to squash it and get it under control again. And that is the key to understanding why the cell can use these types of feedback mechanisms to ensure that it can only move in one direction through the cell cycle and not suddenly back up and change its mind. Okay. All right. Let's go ahead and stop there.